Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Enhancing Treatments Using Targeted Protein Degradation. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank GenScript, who developed the content for this presentation. GenScript is the leading contract research organization in the world providing genes, peptides, proteins, CRISPRs, and antibodies. Since its foundation in 2002, GenScript has grown exponentially through partnerships with scientists conducting fundamental life science research, translational biomedical research, and early-stage pharmaceutical development. The company is recognized as having built a best-in-class capacity and capability for biological research services, encompassing gene synthesis, peptide synthesis, custom antibody and protein engineering, and in vitro and in vivo pharmacology, all with the goal to make research easy. Now, it's my pleasure to introduce our speaker for today's webinar, and he is Dr. Yohang Huang, Doctor in Biochemistry, Protein Senior Scientist at GenScript. Hello there. Dr. Huang has been responsible for the management and technical improvement of the baculovirus insect cell expression platform with his extensive research experience. Dr. Huang achieved his Doctor of Natural Science degree and did postdoctoral research at Free University Berlin. The topic he focuses on is protein biochemistry, which includes protein-protein nucleic acid interaction, protein nucleic acid complex structure, and function analysis. His research findings have been published in research journals such as Molecular Cell, Nucleic Acids Research, and Nature Communications. Now, with that all done, it is now my pleasure to pass the mic and the controls over to Yohang. So Yohang, when you're ready, you may begin.
Okay, good morning, everyone, and welcome. Can you hear me?
Yes, we can.
Okay, then let's start. Today, I will discuss a hot topic in drug discovery strategies, target protein degradation, a breakthrough approach that has the potential to turn previously undruggable targets to be druggable. In this talk, I will elaborate on the following three sections. First is what is targeted protein degradation and the introduction of E3 ubiquitin ligase and the development of UPD drug discovery, and what GenScript offers for UPD drug discovery research. First of all, let's talk about what is TBD. Prior to clinical trials, drugs discovery entails the selection of targets, the majority of which are proteins. This is followed by high throughput screening or docking and structural analysis to identify lead compounds. These lead compounds, I will make a pointer. These lead compounds will undergo several rounds of optimization to finalize their design. Small molecule drugs are among the most crucial methods of disease treatment in present days still. Traditional drug discovery primarily focus on inhibiting the biological function of target proteins, where blocking the functional or catalytic pockets. This strategy requires that the target protein to be accessible, and considerations must be made for affinity, specificity, and activity. There are clear limitations to this approach. For instance, only an estimated of 20% of cellular proteins are predicted to be potentially targetable. Furthermore, issues such as side effects and rapid resistant foundations are also challenges that need to be addressed. To overcome the limitation of old-fashioned designs, scientists are actively exploring new strategies for drug design, one of which is the most promising approaches involve leverage, leveraging the protein's degradation pathway within cells. In eukaryotic cells, proteins can be eliminated via two pathways, the proteasome and the lysosome. In proteasome degradation, in proteasomal degradation, the protein substrate is recruited by E3 ubiquitinase and then following with ubiquitination and subsequently digested by the proteasome. Lysosomes, on the other hand, are responsible for the degradation of surface proteins and large particles. Correspondingly, drug discoveries that target proteins of interest and trigger their degradation, where these two pathways are evolving. Its concept is to use biofunctional molecules to link the target proteins and the degradation machinery together, thereby switch the degradation. Today, we'll focus more on a hotspot in drug development strategy, the ubiquitin proteasome degradation, in short UPD. Currently, there are three kinds of UPD molecules are developed following the UPD pathways. Molecular groups, PROTACs and ABTAC. The molecular group is a bifunctional molecules that itself can bind both to E3 ubiquitinase and the protein of interest to bring them together. And PROTAC is the hetero bifunctional molecules with two functional groups which binds to E3 ubiquitinase and the protein of interest respectively to bring them together. And the most emerging technique, the ABTAC, is using the bio body to bring the protein of interest, the transmembrane or cell surface protein of interest together with the transmembrane E3 ubiquitin ligase to trigger the degradation. Ubiquitin ligase, ubiquitin proteasomal degradation techniques present significant advantages compared to traditional drug development. Firstly, it ignored the constants of the target size, which dramatically expand the list of druggable proteins candidates. Secondly, each of the UPD molecules can participate in several rounds of reactions, resulting in a much lower functional dose. Moreover, the use of intercellular machinery can lead to fewer side effects. Subsequently, the development of resistance is more gradual. In addition, UPD eliminate the subsequent ablation of all the associated functions of the target proteins. Since the TBD is proposed in 1999, the UPD techniques have develops very quickly and reaches a series of milestones. This including the discovery of the protein and molecular groove molecules and and the report of the first VHL or the first CRBN-based protein molecules. And in 219, there's two protex molecules approved to be clinical trials. And 2021, the Pro-AB, which is also the AB tech, is proposed. And nowadays, more and more UPD molecules are entering the clinical trials. E3 ubiquitin ligase are the very important part in ubiquitin ligase ubiquitinations and form an essential components of UPD drug discovery. In the upcoming session, we'll go into E3 ligases and describe about the UPD drug discovery development. Within the ubiquitination complexes, E1 and E2 enzymes are responsible for the ubiquitin transmission, whereas E3 ubiquitin ligase directly recruit the protein substrates to the ubiquitination machinery. This leads to the substrates being ubiquitinated and subsequently captured by the proteasome and degradation. E3 ubiquitin ligase plays a significant role in numerous vitro cellular processes, such as metabolisms, immunity, DNA degradation, DNA damage repair, and also the cell proliferation and apoptosis. There are over 700 of E3 ubiquitin ligase in human, which are apparently divided into three classes based on their structure and the functional characteristics. These three are the RBR E3 ligases, HEC E3 ligases, and the most popular one, the ring E3 ligases. Ring E3 ubiquitin ligase is the largest family of the ubiquitin ligase, boasting a variety of functional architectures such as monomer, dimer, oligomer, and the multi-subunit complexes. Among these, the cooling ring E3 ligase is the most sought after targets. Each of these E3 ligase align with specific cooling proteins to form active complex with conserved structures. Besides their involved in numerous essential cellular processes, ring E3 ubiquitin ligases also demonstrate tissue specificity. A unique proton map of E3 ligase can be identified in different cell groups throughout the body, providing valuable guidance for tissue-specific therapeutic. Since the introduction of UPD, its advantage of target protein scope and dosage, as well as the potential for specificity in tissue and protein substrates, it attracts numerous well-known pharmaceutical companies to enter this field. However, as we can see from the periodic table, although the E3 ligase pool is huge, almost all pipelines shown here are based on just CRBN and VHL, and also the target proteins are most of the target proteins are the kinase. With these backgrounds, new trends in UPD drugs developments are beginning to emerge. Firstly, there are a growing need to expand the pool of E3 ubiquitin ligases and protein target candidates. A significant number of novel E3 ligase candidates, such as those from the DCF, TRIM, MARCH families, are currently being validated in various pipelines. Similarly, the focus on target proteins has gradually diversified it from a strong emphasis on kinases. Secondly, the selection of target E3 ubiquitin ligases is becoming more specific to cells, tissues, and substrates. Besides its application in human therapeutics, UPD also being explored to applicate in agriculture to protect the crops from diseases, weeds, and pests. Yeah, now we have a more clear view of the bright prospect of UPD drug development. In these last sessions, I will show that what are offered in GeneScript to support a UPD drug discovery research. In GeneScript, most of the target proteins, including E3 ubiquitin ligases, are produced using the baculovirus insect cell expression system. This is largely due to the baculovirus vector's high capacity for the gene of interest insertion, because it has a really large genome with 130 kilobase pair. And the BIC system moreover, the BIC system utilized the baculovirus very late promoters. With these promoters, the recombinant proteins expression will express in regular manners. Consequently, the BIC system is particularly well suitable for the overexpression of large proteins harmful to cells, and multicellular complexes, which are typically the typical characteristics of drug target proteins. With the BIC systems, GeneScript has successfully delivered over 200 cases involving more than 20 different E3 ligase constructs with really high quality. We're showing the cases we made here. You can see we can make the CRBN DDB1 complex and the trim and DCAF DDB1 complex and DCAF alone proteins really nicely. And also we have experience with others E3 ubiquitin ligase, for instance, the F-box, the ZNF family ubiquitin ligase, and also as well as KLHL, KLHDCS family E3 ubiquitin ligases. In addition to E3 ubiquitin ligases, protein substrate targets also generate another essential portion of UPD drug development. In GenScript, we also have experience working with various of protein targets. For instance, the kinase, we have delivered over 220 cases with more than 45 different candidates, different kinase candidates, and also for a lot of cancer target proteins, for instance, the polymerases, helicases, and also some transcriptional factors and something like that. And also we have experience with others therapeutic target proteins such as USPs and EGFR something. We also can make it in very high qualities. And here also we show the cases we made before here. And currently we are introducing a guarantee service for several popular E3 ligases and target proteins. Some of them are even unique in the market. We also have an ongoing beta testing program which offers proteins with benefits such as guaranteed yield and purity, quick turnaround time, and cost efficiency. If you're interested, please feel free to reach out to me. Okay, and then yeah, this is all for my talk today. Sonia.
Well, thank you very much, Yohang, for that very interesting presentation. I'm sure everybody enjoyed it. And now we're going to put on our webcams and we're going to start with the Q&A portion of the webinar. So as a reminder to our audience, you still have the opportunity now to send in your questions by using that chat box. So if you can go ahead and send those questions through. And Yohang, if you can just put on your webcam and then we will begin the Q&A.
Yeah.
All right, so I have here the first question that came through quite early. This audience member is asking, what are the uses of this treatment method and can it be used in cell therapy and gene therapy?
Actually, this strategy mainly set up for small molecule and also some cases for the antibodies. So I think it's not suitable for the gene therapy and cell therapy because it's some kind of outside put in, but not from the cell inside. So I think it's not suitable for the gene therapy and the cell therapy. Yeah.
Okay, all right, thank you for that. Okay, let's go on to our next question here. This audience member is asking, why can the very late promoter of baculovirus express harmful proteins at a higher level? What are your thoughts on that?
Because actually, baculovirus insect cell system is a system which utilize the baculovirus. In the life cycle of baculovirus, in the very late stage, the promoter will express in a reckless manner. It doesn't care about the whole cells, and because finally the whole cell will be lysed by the virus, so it will express reckless. So no matter the proteins harmful or not, it will express as it is. Yeah.
Okay. All right. Thank you so much
Mhm.
for that. I'll jump to the next question. This audience member is asking, why is the BIC system suitable for the expression of recombinant E3 ubiquitin ligases and most target proteins?
Um, yes, as I mentioned before, because most of the target proteins, for instance, the E3 ubiquitin ligases, and the for instance some polymerase, their all expression inside the cell is actually sometimes it will have some toxic effect for the cells. For instance, E3 ubiquitin ligase, if the E3 ubiquitin ligase level is really high, it will
Mhm.
degrade the proteins intracellular, and will make the cell growth will make the cell grow have some problem, and will
Oh.
sometimes even kill the cells. And similarly for the other target proteins like the polymerase, it will break the balance inside the cell. So
Mhm.
the cell growth and proliferate and apoptosis, something like that, will totally destroy. So it's harmful. But however, the big system can overcome these effects. Uh,
Mhm.
as I said before, it's used the very late promoters. So in the very late promoters expression, the proteins will the cells will overcome all the harmful effects of the overexpression proteins. Yeah.
Okay. All right. Thank you very much for that very detailed answer. I'm just getting a bunch of questions coming in from our audience members right now. So give me a moment here as I get everything all organized.
Mhm.
Okay, and I'll go to the next question here. This question is, what are the differences between molecular glues and PROTACs?
Okay. Actually, I would say they are very similar, but the molecular group has itself can bind to molecules, can bind to both E3 ubiquitin ligase and also the protein of interest. So it's a small molecule. It
Mhm.
has it can bind two stuff, but for PROTACs, actually it's two functional group linked by a linker, and so they have the bifunction, and it's a chimera. So it's kind of similar, but it's not very similar.
Mhm.
When people develop the UPD drugs, normally the PROTACs will be more easier, because when we develop PROTAC, we can develop the groups based on the single proteins, and don't
Mhm.
have to consider they're not the other one. But for the molecular group, people need to make the molecules bind to both proteins in the same time. So it's a bit more difficult with the molecular group than the PROTAC development.
Okay,
Yeah.
all right. Thank you again for that. I just sent some more questions through there. If you can take a look at them. Here is the next question that I have. Why can the UPD drug molecular trigger several rounds of ubiquitination? Why
Okay.
can the UPD drug molecule trigger several rounds of ubiquitation?
Because the ubiquitination is a reactions which will be a cycle, and each UPD drugs can trigger after the one UPD drug trigger one cycle of the reaction, it will
Mhm.
release from the ubiquitin ligase and then trigger another one. So every one molecule can trigger several reactions, but the traditional small molecule drugs, each of the molecule can just target one protein's target.
Mhm.
So that's why the traditional small molecule drugs will need higher dose, but UPD drugs will need much fewer functional dose. Yeah.
Okay, all right, thank you for that. I'll go to the next question here. Okay, this audience member is asking for the beta testing. Is it guaranteed that if people request some mutations or additional sequence design?
Yeah, actually, we'll guarantee the also guarantee the year and also the purity of the protein. However, for some big change, we need to evaluate first, and moreover, because there's some change in the sequence, so we need some more. We need additional time for the gene synthesis and also some virus generation, something like that. But if people just want the default sequence or the default constructs of the proteins, then it will be much faster. Yeah.
Okay, all right. All right, let's go to the next question here. Let me just get it lined up. Okay. Has there been a clinical trial in the field of E3 ligases for cancer treatment or not?
Yes, I think so. Uh, Actually, we can go to the BTK. Actually, is my
You want,
slide still there
yeah,
or
I'll pass the slides to you. Here, I'll send it to you now.
okay?
There you go. And you can share it.
Mhm. Yes, as I show here,
Hmm.
and as I as far as I concerned, actually some disease like lung cancers and breast cancers, there's pipelines to trigger these cancers with the PROTACs. Yeah.
Oh, okay. All right. Perfect. All
Mhm.
right. I'll take back the controls.
Okay.
Thank you very much for sharing that. And I'll go to our next question. This may be our last one unless we get some other questions coming in. This question is, how can UPD be used in agriculture? And are there any other fields where UPD can be applied? Your thoughts on that?
Okay. And actually, as I know now, there's some research just in agriculture, um, and actually how to say the UPD drugs will use for will target some specific proteins in the wheats, for instance, or in pets or some disease like some virus proteins. So the so the UPD drugs can trigger the um, degradation of these proteins either in the with cells within the with cells or in the Crohn's cells to degrade the virus proteins to protect the crops.
Mhm.
And in other fields, I think for animal health, perhaps people also can apply with UPT drug discovery to develop the drugs for animals. But
Mhm.
now I didn't notice there's any pipelines with the animal health. Yes.
Okay. Hope I'm saying this correctly. This audience member is asking if endogenous expression of targets through CRISPR seems to be favored when measuring protein degradation from PROTACs. It's a topic for expression from transient transfection or stable cell lines, an equally valid method.
Sorry, can you say it
Question number
again?
5. Indigenous expressions of targets through CRISPR seems to be favored when measuring protein degradation from PROTACs. Is the topic expression from transient transfection or stable cell lines an equally valid method?
Um, Sorry, I need to think. I
Yeah,
need to.
go right ahead,
Oh,
and it's right there, number
yeah.
5.
Question 5. I had to. Mm.
Is that a question you would rather
Uh,
answer offline, or are you okay to answer it now? Do we have time for that?
Perhaps I need to, let me see. I need to consider about it.
Hmm.
Mm, I think we I think the transient transvection will be better because if we if it expressed in the stable cell lab, perhaps the whole feature of the cell will be modified because it will express stably.
Hmm.
Mhm.
Okay. All right. Thank you so much for attacking that one. And it was a long one. So thank you very much for that,
Yeah,
Johan. All right. Well,
yeah,
thank you very much for those questions. We have reached the end of the Q&A portion of the webinar. If we could not attend to your questions, the team at GenScript will follow up with you after this presentation. And if you have any further questions, please feel free to submit them to this email address, Johan's email address here that you see on your screen. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive of this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Additionally, I have shared a link in a few moments. I'll share a link to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. So I encourage you to do that. Now join us in thanking our speaker for their time here today, Johan. So thank you. There
thank
he
you.
is. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X Talks, we thank you for joining us. Until next
Thank
time,
you.
please take care. Bye for now. I'm Sonia Hunt. Bye everyone. Bye Johan. Bye
Bye,
everyone.
everyone.